Let's have a look at homework 9, starting with problem 11. This is showing you a peptide made up of 2 amino acids. It's got 5 different areas highlighted that it wants you to identify. So your choices over here, the peptide bond, which is the amide bond, and you have a definition for that in your notes. Hydrophobic side chain, and down here on the bottom, hydrophilic side chain. That is probably going to be something that you want to look up look at your notes for the tertiary structure of a protein. Look up what it means to be a hydrophobic side chain, what it means to be a hydrophilic side chain. So this is going to be stuff from the beginning of chapter 21 as well as kind of stuff from the end of chapter 21. And the amino group and the carboxylic acid group or carboxyl group are, so this should be pretty straightforward for you. Here we have 3 amino acids together, and your job is to connect them into peptide bonds. So it's kind of making it a little bit easier for you because you don't have to draw the whole entire structure of every single individual amino acid. But what you want to do is take these amino acids and modify their structures as necessary. So you got to get rid of the parts that disappear when the amino acids are connected to each other, and then create that new bond in between the two. And so that's all that you're going to be doing. That's all you're modifying there. And you don't need to draw the water molecules that are being formed when this type of thing is happening. Protein secondary structure. So this is asking you questions about the alpha helixes and the beta pleated sheets. And this is specific to the secondary structure. So don't be tricked by questions or possible answers about disulfide bonds that don't exist in the secondary structure. Stop. That's not an option. So you're looking at things that make sense for the secondary structure, the alpha helixes and the beta pleated sheets. Here is a picture of a drawing of a secondary structure, and in green it has 3 possible hydrogen bonding interactions. So it wants you to identify which one of these, and maybe is it more than one, that represents hydrogen bonding in a secondary structure. So first thing you have to keep in mind is, what is a hydrogen bond? Is a hydrogen bond a connection between 2 hydrogen atoms? It is not. A hydrogen bond is a connection between a hydrogen and either an oxygen or a nitrogen or a fluorine. So an HH bond is never a hydrogen bond. And so option A is not an option. And then for the other 2 or 3, B, C, and D, those are all bonds between hydrogens and oxygens, so that's okay. But you have to ask yourself, is that actually where the hydrogen bonding occurs between 2 amino acids in the secondary structure? Look at your notes for hydrogen bonding in a secondary structure and ask yourself what thing is hydrogen bonding to what other thing specifically, because it's not any hydrogen bond, it's a very specific hydrogen bond. And then here's question 15, looks almost exactly the same. It looks almost identical. Question 16. This is asking about a tertiary structure. So now we're looking at tertiary structures, and it wants you to identify, well, the disulfide bond, that one should be easy because you drew that in your notes. You have hydrophobic, and so in your tertiary structure you have what does it mean to be a hydrophobic interaction. You have electrostatic, which we also called hydrophilic, so look in your notes to see what that means. And then you have a hydrogen bond, like a full-on hydrogen bond is a possibility as well. So I feel like you can match those up as long as you know what part of your notes to look at. And then the last one that you're looking at here is question 17, and this is asking you some different things and are giving you some different things and asking you to sort them to what part of the protein structure does that belong, like the alpha helix and the beta pleated sheet. Those are both secondary structures. So the overall shape of a single polypeptide unit, that's saying the overall 3-dimensional shape when it scrunches itself all together. What does that look like? an overall macromolecule structure containing more than one polypeptide. So that's what we would refer to as multiple proteins that have like hydrogen attracted to each other, hydrophilically or hydrophobically attracted to each other, multiple proteins attracting to each other. And the order of amino acids, the sequence of amino acids. So those are some hints to help you with that problem and with all of these problems.